this set of videos, I am going to describe or summarize a very important article with respect to the research we do in the Hammond Lab here at Illinois State University. That article is Turner and Perkins from 1979 in the journal Genetics, volume 93, and it starts on page 587. So in this article, what Turner and Perkins describe is the discovery of 3 Neurospora spore killers. So Neurospora SK1, SK2, and SK3. Now that's for spore killer 1, spore killer 2, and spore killer 3. And essentially, so what are the Neurospora spore killers? So we can consider them to be selfish genetic elements. So as an example, let's assume we isolate a Neurospora. Let's pick intermedia. Now this is a species of the fungus Neurospora. Let's say we have an isolate from the wild, and let's say this isolate is what we would call SK2K. Now that means this isolate here has all of the genes required to perform SK2 type spore killing in certain crosses. As an example, let's take a different isolate. So Neurospora intermedia. Let's call this isolate 1. This one's isolate 2. Now let's say this isolate has the SK2S genotype. The S means it is susceptible to spore killing by an isolate with the SK2K genotype. So it is susceptible. Now what that means is when we perform this cross, and if you watched the introductory Neurospora biology video, you would know that these have to be opposite mating types. And if these should mate in nature, or we perform this cross in the laboratory, what we should get is the formation of fruiting bodies, which are the perithecia, right? This is a single perithecium. Inside the perithecium, there should be 200 to 400 asci, which are the spore sacs, each spore sac resulting from a single meiotic event. The meiotic event resulting from a nucleus donated from this isolate and one donated from this isolate. And each individual ascus should look something like this. And we should see 4, instead of 8, we should see 4 darkly pigmented ascospores and 4 shriveled up dead ascospores. So these, we consider these viable, these are inviable, and nearly all of the time, let's say this is a very efficient process, this spore killing process, and 99.5% or more of the time, these ascospores have the SK2K genotype. So if you remember from your introductory genetics courses, Gregor Mendel would say, right, well, half of the offspring should inherit these genes, and half of them should inherit these genes. But the Neurospora spore killers, the way they work, they eliminate nearly all of the offspring that do not inherit the genes for spore killing, right? So 100% of the offspring coming out of this cross right here inherit these genes. And that's why we call these selfish genetic elements. So Turner and Perkins discovered SK1, SK2, and SK3. They discovered SK1 in a species called Neurospora Cytophila. Now they discovered SK2 in a species called Neurospora intermedia. They also discovered SK3 in that species. So currently in my lab, we focus just on SK2 and SK3. And for the undergraduate project in spring and fall of 2023, we are just focusing on SK3. So I'm going to cross this one out for now. In the next videos and the upcoming information I'm going to give you is going to refer mostly just to SK2 and SK3. So another thing they discovered was that in N. intermedia, in N. crassa, well, Neurospora intermedia in Neurospora crassa, they discovered isolates in each species that were resistant. So resistant to spore killing, but not spore killers themselves. And this finding actually turns out is going to be really important for making sort of big advances in our understanding of how spore killer works. And that a description of this will come a little bit later. Okay, so now I'd like to go over some of the important spore killer phenotypes that we will need to understand as we go forward with our various projects. So let's start with the basic spore killer 2 killer isolate cross to spore killer 2 sensitive isolate. And we already went over this one, but to make sure we're all on the same page, I will quickly diagram what an ascus. So if we perform this cross right here, SK2K cross to SK2S, and they form parathecia. Inside the parathecia, we will see 200 to 400 asci, and those asci should have the following phenotype. So 4 black viable ascospores and 4 shriveled, what we call white, which they're not really white, but they're translucent. But we call them white. So 4 white ascospores, which are inviable. So that would be the 4 black, 4 white phenotype, which is sometimes written like that, 4 black, 4 white. So next, what we have is, let's say we do a cross between a strain that is SK2K, so it is capable of killing SK2S strains. So let's say we cross that to another SK2K strain, so of opposite mating type, right? We can perform this cross as long as the isolates are of opposite mating type. And so when that happens, and this is what Turner, all of these things, this is what Turner and Perkins discovered, right? We're covering the basic phenotypes from the Turner and Perkins 1979 paper. And what they discovered was that all of the ascospores, all 8, are black and viable. So no spore killing occurs when both of the parents of the cross have the SK2K genotype. So 8 blacks, 0 white. Now the same thing occurs with SK3K crossed to SK3S. Now often, so this is a spore killer 3 killer. This is a SK3 killer isolate. This is a isolate that is sensitive to SK3 spore killing. Now often the SK3S and SK2S isolates are the same, but we're only specifying the SK3 susceptibility in this cross. So often we'll do something like that. So in this type of cross right here, if we look at the asci, what we will see, what we should see, is 4 black ascospores and 4 inviable shriveled up ascospores. So we see the 4 black, 4 white phenotype. So we'll call these are the various ascus phenotypes that we should see in these various crosses. Now, for completeness, we will take a look at what we see when we cross SK3K, cross to SK3K, so two SK3 spore killers cross to one another. And again, if they are opposite mating type, we can do this. What we will see, what Turner and Perkins saw, is 8 black viable ascospores. Okay, so no spore killing occurs. So apparently, the spore killers SK2 and SK3 can kill spores when they're mating with sensitive strains, but not when they're mating with a strain of the same genotype with respect to spore killing. And my ascus is a little too big here. Sorry about that. 8 black, 0 white phenotype. Okay, so now let's take a look at what happens when an SK2K is crossed to SK3K. So think about that for a moment. The asci aren't bigger in these crosses. I am not diagramming these to scale. So what they found was that they essentially see 8 dead ascospores. So 0 black, 8 white. Everything dies. Everything dies when a spore killer SK2K isolate is crossed to an SK3K isolate. So the spore killers essentially kill each other, and there's essentially no survivors. So in all of these crosses, this is biology, and so we almost never get 0% survive. We might have a few ascospores produced, and so those ascospores, we say they escaped the killing process. Sometimes just they got lucky. It seems they just got lucky. So we have some others to go over. So SK2K crossed to SK2R. Now, this is a strain that is resistant to spore killing, but is not a spore killer itself. So an SK2R strain is resistant to spore killing by SK2K, but it is not a spore killer. And so if we look at these asci, we should see 8 black viable ascospores. 1, 2, 3, 4, 5, 6, 7, 8. And I'll color these in for completeness. So 8 black, and half of the offspring are going to have this SK2K genotype. They're going to be spore killers, and half are going to have this genotype, SK2R, the resistant genotype. So they won't be spore killers, but they will be resistant to SK2K based spore killing. Same thing goes for SK3K strains. When they are crossed to a strain with the SK3R genotype, everything survives. 8 black ascospores. Half of the ascospores will have the SK3K genotype. They will be spore killers of the SK3 type, and half will be resistant to SK3 based spore killing, but they will not be spore killers. So they will not be spore killers. What does that mean? So essentially what that means is if we take this SK2R strain here, and instead of crossing it with SK2K, we cross it with SK2S up here, what we see is 8 black viable ascospores. No spore killing occurs. Half of these will have the SK2S genotype. The other half will have the SK2R genotype. So no spore killing is going on. Half of the offspring are inheriting the genes that make a spore killer, make an isolate sensitive to SK2 type spore killing, and half of the offspring inherit the genes that make an isolate resistant to SK2 based spore killing. And the same thing would occur if we did the cross between SK3S and SK3R. All the ascospores would survive because there's no spore killer in the cross. Okay, so spore killer 2 and spore killer 3 were identified by Perkins and Turner in the species Neurospora intermedia. And interestingly, they didn't discover many isolates with the spore killer 2 killing ability and the spore killer 3 killing ability. I think it's only one isolate ever was discovered of the SK3 type, and only maybe 4 or 5 isolates of Neurospora intermedia that were of the SK2 type, and it could be that these were going extinct. We're not really sure. They could be extinct right now in nature. We don't really know. But they didn't discover many of these isolates out in nature, and it could be because resistance built up. Those SK2R and SK3R genotypes built up in the population and took away the fitness advantage that spore killing had when resistance wasn't in the population. So we don't really know. But the important thing is we want to understand how spore killing works. And Turner and Perkins also wanted to understand how spore killing works. And Neurospora intermedia is not the genetic model species of Neurospora, right? That's Neurospora crassa, which has been a genetic model studied by geneticists since like the 1930s, 1940s. So Neurospora crassa is the genetic model. Luckily for Turner and Perkins is that these are so closely related that you can actually perform crosses between them in the laboratory, and maybe they mate at a very, very low level in nature. I mean, that's a topic for a population biologist. And there's this definition of a species which says, well, members of a different species can't mate and produce offspring, and that definition of the species falls apart when we talk about fungi like Neurospora, where researchers have identified different species or classified different species, defined different species, and we can get them to mate in the laboratory. Mating doesn't work well. They don't produce very many offspring, but it allows researchers to move genes, say the genes that control SK2 base spore killing and the genes that control SK3 base spore killing in intermedia, and move them over into Neurospora crassa for studies that are just easier to do with Neurospora crassa because it's well known. It's a better known organism than Neurospora intermedia, and there are a lot of tools for doing genetic analysis with this species. And so what they did to move these genes from intermedia, the genes that cause SK2 to be an SK2 strain and the genes that cause SK3 to be an SK3 strain, they moved them through back crossing. Okay, so how does that work? Okay, so how did Turner and Perkins do their back crossing? So let's take Neurospora intermedia, let's take an SK2K isolate. Now we want to move all the genes that are required or that make an SK2K isolate and SK2 isolate. So we're going to move all these genes for spore killing into Neurospora crassa. And so essentially what they did was perform this cross, an N intermedia isolate, let's say of one mating type, N crassa isolate of the other mating type. This is the SK2K genotype. This is the SK2S genotype. They're able to get just a few, say, ascospores from a parathecium, right? There should be thousands of ascospores, but this is an interspecies cross. It doesn't work that well. They get a few offspring of, let's say, one viable offspring. They should have the SK2K genotype, right? Assuming spore killing is working. But other than that, how much of the genome is going to be inherited, let's say, from the N intermedia parent? And how much is going to be inherited from the N crassa parent? Should be half from each, right? So we inherit half of our DNA from our mothers and the other half from our fathers. Now, same thing happens in fungi. So an intermediate genome and 50% N-crassa genome. And the genes for SK2K are there. Now, so what we want to do, the goal is to increase this 50% towards 100%, raise this towards 100%, and decrease this 50%, decrease that towards 0%, right? Get as low as possible towards 0, while keeping all the genes required for SK2 based spore killing. And the standard way to do that is to just do this very similar cross, the back cross, this offspring with this parent right here. So this offspring right here, which is a hybrid now between intermedia and N-crassa, we do this cross, N-crassa, SK2S, and now probably getting more offspring now, right? Because the genomes are getting more similar. And let's just take one offspring. Assume it has the SK2K genotype, it has all the genes required for spore killing. And half of the offspring half of the genome came from this parent, right? And half came from this parent. So what we're going to have to do is this parent right here, instead of is going to give half of its N-intermedia genome to the offspring and half of the N-crassa genome to the offspring. So 25% is going to be N-intermedia, and essentially it's getting 25% N-crassa from here and 50% N-crassa from there, so 75% N-crassa. Okay, so we're getting even closer. We're getting, the genome is almost getting closer and closer to 100% N-crassa. And so we do this time and time again. Every time we're cutting the percentage of N-intermedia DNA in the genome by 1/2, and increasing the percentage of N-crassa DNA by the equal amount. So the next time we do this cross to N-crassa SK2S, we are going to get an offspring that is SK2K, 12.5% N-intermedia genome, and 87.5% N-crassa genome. So essentially what they did was they moved the genes for SK2K spore killing and SK3K spore killing from N-intermedia into N-crassa by doing many, many back crosses. And eventually what they have after they're done is they have N-crassa strains, N-crassa isolates that have the SK2K genotype and the SK3K genotype, so they're able to study or study the genes that cause spore killing better, because they have these genes working, or they have the genes into an N-crassa genetic background, we will say. So crosses now work very well with other N-crassa strains, let's say of the SK2S or SK2R genotype, or SK3S or SK3R genotype. So all the studies can be done in N-crassa now because of this back crossing, with the availability of these isolates that have nearly 100% Neurospora crassa genomes, except for the genes required for SK2K or SK3K spore killing. Okay, so after Turner and Perkins introgressed SK2 and SK3 into Neurospora crassa from N-intermedia, what they were able to find is that the genes for spore killing are located on chromosome 3. So they're located on chromosome 3. And they also found something interesting. So there is a recombination block, or suppressed recombination on chromosome 3 when SK2 and SK3 are crossed with a susceptible or a sensitive strain, a strain that is sensitive to killing by either one of these. Okay, let me go over that in more detail. So to show this, they used 3 genes that are also on chromosome 3. We're going to call these genes A, L, and H. Now these genes come in this cross in 2 different alleles. So A plus and A minus, L plus and L minus, H plus and H minus. So in this cross, that in one of the crosses they did, they have the Neurospora crassa SK2K isolate that has the A plus, L plus, and H minus alleles for these genes. And again, these genes are on chromosome 3 along with the genes required for that allow a strain or cause a strain to be a spore killer of the SK2 type. And so they cross that with an SK2S strain that's sensitive to spore killing, and this strain has the opposite alleles for each of these genes, A minus, L minus, and H plus. Okay, so let's go back to our introductory genetics. So at the beginning of meiosis, we have chromosome 3 from this parent that's going to pair up with chromosome 3 from that parent in the meiotic cell. And so let me diagram chromosome 3 from this parent over here, SK2K. So this is a single chromosome that exists as a pair of sister chromatids that share a common centromere. And that's how that's what the chromosomes look like at the beginning of meiosis. And so I'm going to put A plus here, L plus here, and let's say H minus here. Okay, now that is chromosome 3 from the SK2K parent. Now I should diagram these a little closer, but I need room to label the chromosomes, so I'm going to diagram this chromosome 3 from the SK2S parent over here. It's going to be A minus, L minus, and H plus. Okay, so in all of the meiotic cells that are formed in the perithecia resulting from a cross or a mating event between SK2K and SK2S with these genotypes, the chromosome 3s are going to pair up, and that's going to allow crossing over to occur between let's say between A and L here and between L and H here. So we're going to be able to get a physical exchange of DNA between these chromosomes. However, interestingly, Turner and Perkins saw 25% crossing over between these 2 genes, L and H, and 0% crossing over between A and L. 0% crossing over between A and L, which is strange, because in control crosses, when spore killer SK2K is not part of the cross, they usually see about 10% to 11% crossing over between these 2 loci, between the A gene and the L gene. So this data suggests there's some sort of recombination block or recombination suppression going on. No crossing over is allowed between these 2 genes. So this was the first evidence they have for that. They did the same thing with SK3. They did the same thing with SK3. And so all we need to do is change this 2 to a 3 and this 2 to a 3. Everything else is the same. And what they found, again, was 0% crossing over between the A and the L. And they saw 11% crossing over between the L and the H. So 11% crossing over. And so how do they determine the percentage crossing over? What they do is they look at the offspring. They look at the offspring, right? And they count the number of offspring that match the parent, let's say for L and H. If the offspring is L minus and H plus, well, no crossing over happened to form the genotype of that offspring. If it's L plus and H plus, oh, then we must have had a crossover. They must have had a crossover during the meiotic event that led to this offspring right here. So essentially what they do is they collect offspring and look at the phenotypes and determine what the percentage of, they determine the percentage of crossing over that must have happened between each pair of these 3 genes. Okay, so again, they didn't see crossing over between A and L. Now let's just look at the control cross so I can emphasize this point. Their control cross that they did, so was they just took SKS strains, either SK2, it doesn't matter, they're always the same, right? So I've been labeling these SK2S for sensitive to SK2 and SK3S for sensitive to SK3, but essentially it's just the same strain. It's just sensitive to both. And so let's just label these as SKS crossed to SKS, chromosome 3 from this strain, chromosome 3 from this strain. Let's assume this strain is A plus, or not, no, let's assume, let's show the genotypes that they use in this cross. So they use A plus, L plus, and H minus, cross to A minus, L minus, H plus. I'll diagram the chromosome 3s, put them a little further apart this time. Oh, I put L down here, right? So A plus, L plus, H minus, A minus, L minus, H plus. And what they saw was that 20% crossing over occurred in the offspring. Looking at the offspring, what they were able to conclude or determine was that 20% of the time we got crossovers between L and H, and 11% of the time we got crossovers between A and L. 11% of the time. So it's a big contrast with 0% right in the spore killer crosses. Okay, so these results suggested that when you have SK2K crossed to SK2S or SK3K crossed to SK3S, that there is a recombination block or recombination suppression for at least part of chromosome 3. And then in the next video.